Hi everybody, so today I'm going to talk about energy dependent transport across membrane plasma. I will start off with the basic mechanism of active transport. Basically, there's three main ideas that you have to know regarding the mechanism of active transport, which is first, the solute must move against concentration gradient, which means the solute must move from a lower concentration to a much more higher concentration. And this active transport require energy, and it uses transporters, or also known as pump. So we move on to active transport. Active transport have two types, which is primary active transport and secondary active transport. For primary active transport, like I said before, each active transport requires an energy, and so does primary active transport. It requires energy in form of ATP. The hydrolysis of ATP is by a transporter that will provide the energy for primary active transport. The transporter is actually an enzyme called an ATPase, and ATPase will catalyze the breakdown of ATP, and in the process phosphorylates itself. What does it mean by breakdown of ATP is that it will phosphorylate ATP into ADP and 1-phosphate organic that will change the conformation of our transporter, and this will also increase the affinity of our transporter to a selective solute. The main function of primary transport is to maintain cell's potential, and there are four major primary transporter, which is sodium potassium pump, calcium pump, proton pump, and proton potassium pump. Next, we'll move on to secondary active transport. Secondary active transport also requires energy, but not in form of ATP. It requires energy that derived from electrochemical gradient, and this electrochemical gradient is provided by our primary active transport. So you can see that secondary active transport and primary active transport is actually very related and dependent to each other. Secondary active transport is different from primary active transport by its use of an electrochemical gradient across a plasma membrane as its energy source. In primary active transport, as I have explained earlier, it requires energy in form of ATP, which will then phosphorylate the transporter into ADP and inorganic phosphate. In secondary active transport, the movement of an ion down its electrochemical gradient is coupled to the transport of another molecule as well, such as amino acid and glucose. There are one example that I can show you here. What does it mean by the coupling of the transport of another molecule? Here you can see the sodium dependent secondary active transport is actually can be divided into two types, which is sodium calcium counter transporter and sodium glucose co-transporter. Counter transporter means that the movement of sodium is opposite to the movement of calcium. For example, sodium will move into the cell, and the calcium that will move together with the sodium, but in a different direction. So if sodium move in, calcium will attach to sodium, will couple with the sodium, but it will move out from the cell. But for co-transporter, it means that the ion is moving in the same direction as the sodium, which means both of sodium and glucose will couple together and move, let's say for an instant, outside of the cell. Okay, I have been talking about this electrochemical gradient, but what it is actually? Electrochemical gradient is caused by uneven distribution of ions, and it is the combination of electrical and chemical gradient. The electrical coming from the charge of our ion, like sodium plus and chloride negative, calcium 2 plus. That what will make the electrical part, and the chemical part is where there's an involvement of chemical. Besides that, electrochemical gradient can also achieve an equilibrium potential, which we call as Nernst potential. So, beside active transport, we also have vesicular transport. There are two types of vesicular transport, which is endocytosis and exocytosis. Endo simply means into. Therefore, endocytosis meaning movement of substances into the cell. There are two types of endocytosis, which is pinocytosis and receptor-mediated endocytosis. Pinocytosis simply means a cell drinking. Why is it so? This is because the vesicle in pinocytosis will encloses a small volume of extracellular fluid to bring the extracellular fluid into the cell. This process is non-specific because this vesicle simply engulfs the water in the extracellular fluid along with whatever solids are present. That's why it is unspecific. This solid may include ions, nutrients, or any other small extracellular molecule. In contrast to fluid endocytosis or pinocytosis, receptor-mediated endocytosis is a very specific process because it have receptor that must bind to a certain ligand in order to enter the cell. There are receptors that is bounded on the plasma membrane that will recognize one ligand with high affinity. In one form of receptor-mediated endocytosis, the receptor undergoes a conformational change when it binds with the specific ligand. Through a series of steps, a cytosolic protein called clathrin is recruited to the plasma membrane. Clathrin forms a cage-like structure that leads to the aggregation of ligand-bound receptors into a localized region of membrane, forming a clathrin-coated peak, which then invaginates and pinches off to form clathrin-coated vesicle. In this way, there are a very specific process going on that only a certain molecules can enter the cell. For an example, a cholesterol. So next, we will discuss about exocytosis. Exo simply means out. So exocytosis means movement of substances out of the cell. There are two functions of exocytosis. First is to provide replacement material for plasma membrane, and two is as the route to export large lipophobic molecule, which means you want to export something out of the cell, but it does not permeable, or it cannot diffuse. It cannot diffuse into our plasma membrane because it's a lipophobic molecule. So we will use the exocytosis mechanism to transport all these lipophobic molecules. The example is for excretion is nitrogenous waste, and for secretion is hormone, enzymes, antibodies, and mucus. This secretion means that the cell will secrete these four types of substance into the blood cells. So that's what I mean by secretion. The secretion is not into the cell, but instead outside of the cell into our bloodstream. Thank you.